environment at the University of Nanjing in China. And his research interests include heavy metals in soils and sediments. And he has a special interest in metals which heavily impact on human health, including mercury and cadmium. He's published some 35 journal papers. Most recently, I've seen 2 in Chemosphere on mercury and cadmium, and another in environmental pollution on mercury. 2 of these are focused on paddy soils and understanding uptake of mercury in rice. And today he's going to be talking about remediation of mercury-contaminated soils. So I'll pass over to Huan.
Thank you, Margaret. So hello, everybody. So I should say good morning for people in North America, and good afternoon for people in Europe, and good evening for people in Asian countries, right? So today I'm going to talk about something, or some story about remediating the mercury-contaminated soils. And what's the difference of our story is that we focus on controlling mercury bioavailability instead of controlling the mercury concentrations in soil. So let's start. So before I start my story, there's 2 things, actually 2 theories I want to emphasize. And these 2 are bioavailability and mercury. So first I will tell you why we care about bioavailability. I know that some lecturers have already introduced about bioavailability, but there's something different I want to show. And second, I will show you so why we're concerned about mercury, because not everybody is very familiar with mercury, although I am. So first, bioavailability. So what is bioavailability? So there's one thing bioavailability tells you is that presence is not equal to availability. That means not all the contaminants that is present in the environment could be available for the organisms, or including our human beings. So that means the external dose of the contaminants in the environment is not equal to the internal dose of contaminants in our body or within any organism. But actually, the external dose is much higher than the internal dose of contaminants within the organisms. This is because the bioavailability of contaminants are less than 100% for all the contaminants in the environment. Or we can say that not all the contaminants are available to organisms. So that is how bioavailability tells us. So what's the reason that bioavailability is not equal to 100%? Generally speaking, there are 2 reasons. First is because the interactions between the contaminants and the environment medium. For example, let's think about the metals in the soils. So metals do not exist lonely in the soil, but it always has some kind of binding with the soil component. For example, metals could be bind with the organic matter, or bind with clay, or bind with iron and manganese oxide, or something else. So it's not alone. So it always has some kind of interactions between the contaminants and the environment medium. And that's why the contaminants in the environment is not 100% available to the organisms. So this is the first reason. And the second reason is that so our human beings or any organisms is like a castle. So the door is not always open. So that means not all the contaminants cannot just easily go into the castle or go into this city of organisms. So sometimes the door is closed, and sometimes this door is selective. That means not all the contaminants in any form can enter into this castle or into this city of human beings or organisms. So for these 2 reasons, that bioavailability of contaminants is not 100%, and actually is lower than 100%. So that means normally we cannot see this kind of relationship, because we normally believe that the external dose is linear related with the internal dose, but actually it's not. So normally what we see in the environment is like this. There is no linear relationship between the internal dose of contaminants and the external dose of contaminants in the environment. And if you think about it, actually the slope of these curves are the bioavailability of contaminants to the organisms. So if the contaminant is highly available to the organisms, then we have a larger slope on this curve. But if the bioavailability of the contaminant is 0, then it's just a flat. It's just a zero slope of this curve. That means the contaminant is not bioavailable or 0 bioavailable to the organisms. So why are we concerned about bioavailability? It's because even with the same concentration in the environment for the contaminants, if the bioavailability is high, then we will see a higher concentration of higher bioaccumulation of the contaminants in the organisms, or higher toxicity of the contaminants. And it may need a more urgent remediation. For example, we have 2 paddy soils with the same concentration of cadmium. The paddy soil A has a much higher, perhaps 80% of bioavailability, and the second paddy soil B has about like 10% of cadmium bioavailability to the plants. So definitely, it's more urgent to remediate the paddy soil A because of the higher bioavailability of cadmium in the paddy soil. So this is why we're concerned about the bioavailability. So second, let's talk about mercury. So why are we concerned about mercury? So actually, I just searched the Web of Science and look at the publication about mercury per year in Web of Science. So in this slide, you can see that our interest in mercury, actually the publication about mercury increased very fast in the last 50 years and increased very fast also in the recent years. So also concerned about the publication of mercury in ESTNT, which is one of the top journals in environmental science. You can see a very fast increase in the publication per year in the last 20 years. So that means, although the Minamata Bay disease has passed about like 60 years, our interest about mercury just keep increasing. So why? So have you ever think about why we are concerned about mercury in the environment and why so many researchers all over the world are interested in mercury? So actually, there are several special things about mercury. The first special thing about mercury is about the global transportation of mercury. I think everybody knows that mercury can be reduced to elemental mercury, and elemental mercury is highly mobile, and it can just transport in the atmosphere to every part of the world, to the north, to the south, to the peak of the hill, and to any corner of the world. So that is what's special about mercury. But compared to other metals, for example, cadmium, or some other metals like zinc or copper, so normally it's just a local problem because it will stay there maybe forever. But mercury is not. It can transport. It can transport to different places. So this is the first special thing about mercury and why we are concerned about mercury. And the second special thing about mercury is that mercury can be methylated into one special species called methylmercury. And what's special about methylmercury is that methylmercury is highly bioaccumulative and is highly bioavailable. So thinking about the trophic transfer of mercury through the food chain. So from the bottom of the food chain, for example, in the aquatic system, to the top level of the trophic transfer, for example, in the fish or in human beings, it could biomagnified for about 100,000 times. So that means if you get 1 unit of mercury in the water, and you may probably get about 100,000 units of mercury in human beings or in fish. So that's terrible. So even the low level of mercury in the environment may result in a very high concentrations of mercury in the organisms or in human beings. So that's a terrible thing about biomagnification of mercury. And this is especially true for the species methylmercury. So this is the second special thing about mercury. And the third thing everybody knows is toxicity. So we all know that the Minamata Bay disease in Japan, which poisoned over 100,000 people there. So it's a very terrible disaster in Japan, and it's because of the toxicity of Miss Mercury. So this is the problem of mercury, and this is why we're concerned about mercury in the world. So we just remind you again, why we're concerned about mercury. First is because of the global transportation of mercury to every corner of the world. And the second is about mercury can be methylated into one species called Miss Mercury. And Miss Mercury is highly bioavailable and could biomagnified through the food chain. And the third is about the toxicity or the high toxicity of mercury to organisms and to human beings. So this is why we're concerned about mercury. And this is why we want to remediate the mercury in the environment. And one thing special about the mercury bioaccumulation is that recent study showed that there are several hotspots of mercury in the environment. And one thing special is that called paddy soil, where the rice grows. So in the paddy soil, the environment will favor the transformation of mercury to the highly toxic and bioaccumulative species, Miss Mercury. And then in that case, Miss Mercury can be highly accumulated in rice plants. And you know that rice is a major food in Asian countries. It's a staple food in many countries in Asia. So the bioaccumulation and the methylation of mercury in the soil rice system may pose a risk to human health, especially in Asian countries. So this is why we want to remediate the mercury in the soils, and especially in the farming soils. So let's go back to this figure. So there are two processes that controls the bioaccumulation of Miss Mercury in the plants or in the crops, especially in rice. So the first step is called methylation. And this is mainly controlled by the microbial methylators, or you can see the bacteria. And generally speaking, there are several categories of bacteria that is responsible for the methylation process of mercury. The first category is called sulfur-reducing bacteria. And the second group is called iron-reducing bacteria. Of course, there are some other categories of bacteria that is responsible for the microbial methylation of mercury into Miss Mercury. So definitely, this process of methylation will control how much Miss Mercury could be found in the soils. And the second process that controls the Miss Mercury bioaccumulation is the bioaccumulation process itself. That means how much Miss Mercury in the soil can be taken up by the rice plants or the crops and being accumulated in the rice plants. So it's very simple that these two processes, the first methylation process and the second, the bioaccumulation process, will control the risk of mercury in the soils, in the farming soils. So now we have two choices. The first choice is to reduce the mercury methylation process. Then we can reduce the Miss Mercury concentrations in soils. And the second choice, we can just reduce the bioaccumulation process of Miss Mercury to the crops. So these are the two choices we have. But unfortunately, these choices are not very common in the practice of soil remediation of mercury in the real world. So the very common technique for remediating the mercury-contaminated soils is to remove the total mercury in the soils. So this is normally what people did in the soils. So that means what they did is to eliminate or reduce the total mercury concentrations in soils, and they think that this will subsequently decrease the Miss Mercury concentration in soil and decrease the Miss Mercury accumulation in the rice plants. But the problem of this choice is that it's not environment friendly. So this is a machine that can help you to eliminate the total mercury in the soils. So we just take a look at this machine. So what they did normally to eliminate the total mercury concentration in the soil is to heat the soil to about like 600 degree or maybe 700 degree. And this will just evaporate the mercury in the soils and decrease the mercury concentrations in the soils. So the problem with this choice is that, first, consider the cost. So normally the cost for this method is very high because it needs a lot of energy to just heat the soil. And second, just think about it's farming soil. So after heating at such high temperature, for example, 600 degree or 700 degree or 800 degree, it will just kill all the microbes in the soils and evaporate the organic matter in the soil. So that means in a very, very long time, these soils, after the treatment with heating, cannot be used for farming. So this is very unfortunate because what we want to remediate is farming soils instead of industrial soils. So this is the second disadvantage. And the third is efficiency. This method can just reduce the total mercury concentration in the soils, but it will give no promise that it can decrease, especially the mixed mercury concentration in the soils or the bioavailability of mixed mercury to the crops. So the efficiency of this method to control the mixed mercury bioaccumulation could not be very high. So these are the disadvantages of heating the soil to reduce the mercury concentration in soils. So is there any questions? You can just speak or you can just send a text to me. So this is the first choice. So we see the disadvantage of this method. So actually we have a better choice. As I shown before, instead we can just decrease the mercury methylation process and decrease the mercury bioaccumulation process. So just like the green berry I showed in this slide, to stop the mercury methylation process or to stop the mercury bioaccumulation process. So how about this choice? So in this choice, our main idea is to control the mercury bioavailability, or we can say to decrease the mercury bioavailability. For the first process, we want to decrease or reduce the availability of mercury to microbial methylator. After that, we can just decrease the microbial methylation process and to decrease the mixed mercury concentrations in the soil. So this is the first process. Sorry, Margaret, can you see my mouse on the screen? So if I just point at the slide, so everybody can see it, right? Okay, thank you. So that's better. So for the first process, we want to control the availability of mercury to the microbial methylators, that means the bacteria. And then we can just reduce the mercury methylation in the soils. And for the second process, we want to reduce the availability of mixed mercury to the plants and to reduce the bioaccumulation of mixed mercury in the plants. So this is our second choice. So in the next few slides, I want to show our recent progress in using these two methods to control mercury bioavailability and to reduce mercury bioaccumulation in the rice plants. So for the first process, we tried one very special element, which is called selenium. So I'm not sure how many people are familiar with selenium, this element. So actually, there have been so many studies investigating the interactions between mercury and selenium in the aquatic systems. And those studies have shown that selenium could reduce the mercury bioaccumulation in the aquatic system. For example, it can decrease the accumulation of mercury in the fish or in the deposit feeders or in other aquatic organisms. And this is because of the very strong binding between mercury and selenium. So you can think that selenium just fixes mercury in the environment because of their very strong binding. And the underlying mechanism of the what we call the Miss-Mercury selenium antagonism in aquatic environment, that means selenium decrease the Miss-Mercury accumulation in the aquatic system, are two reasons or two underlying mechanisms. The first one is people think that selenium could facilitate the Miss-Mercury demethylation process. So that means selenium could decrease the Miss-Mercury concentrations. And the second is because of the complexation between Miss-Mercury and selenium. So that means the complexation or the strong binding between Miss-Mercury and selenium would make Miss-Mercury less bioavailable to the organisms, and that decrease the availability of Miss-Mercury to the organisms. So for these two reasons, people think that selenium could reduce the Miss-Mercury availability to aquatic organisms or decrease the bioaccumulation of Miss-Mercury in the aquatic environment. However, there are only a few studies investigating the interactions between Miss-Mercury and selenium in the terrestrial systems. So there are several typical studies. So one study in 2012 investigated the relationship between mercury and selenium in the mining contaminated area in China. It's one of the biggest mercury mining areas in the world, which is called Huan Zhong mercury mining areas in Guizhou province in China. So in that paper, scientists found that there is a very interesting relationship between the mercury concentration in the rice grains, as you can see from here, the y-axis, and the selenium concentrations in the rice grains. So it seems that the selenium in the rice plant can reduce the inorganic mercury concentrations in the rice plants. So from that moment, people just think about the possibility that selenium could also reduce the mercury bioaccumulation process in the terrestrial system, especially in soil crop system. And further evidence is that people found that when you add selenium into the soil, we found a decrease in the Miss-Mercury concentration in the plants here. And also in 2014, people also found that when you add selenium into the soils, you see a decreased Miss-Mercury concentration in the rice plants. So these are the pioneering studies that show the possibility that selenium could reduce the Miss-Mercury accumulation in the plant in terrestrial system. But at this moment, people do not know what's the underlying mechanism of this Miss-Mercury selenium antagonism in soil rice systems. Actually, the reduction of Miss-Mercury concentrations in rice plants could be due to two reasons. The first, it may be because of the interactions between Miss-Mercury and selenium within the rice plants, which inhibit the transportation of Miss-Mercury from other part of the rice plant to the rice grain. So this is the first hypothesis, which is called the in-plant Miss-Mercury selenium antagonism. The other possibility is that the reduced Miss-Mercury concentration in rice grain is because of the interaction of Miss-Mercury and selenium within the soils. So previous studies cannot show which mechanism is mainly responsible for the reduced Miss Mercury concentration in rice grains under selenium amendment in soils. So what we did is we compared two ways of selenium application into the soil rice system. So for the first method, we just spray the selenium onto the leaves of the rice plant. So in that way, we can see the elevated selenium concentrations in the rice plants, but not in the soils. And for the second method, we add the selenium into the soils. So in that case, selenium will be taken up by the root of the plants and go into the above ground tissues of the rice plant and also go to the rice grains. So if we can compare the dynamics of Miss Mercury in rice plants under selenium amendment in these two methods, which is called the foliar application and the soil application of selenium, then it will help us to see which mechanism is mainly responsible for the antagonism between Miss Mercury and selenium in soil rice systems. So interesting, our results show that after we add the selenium into the soils, we see a sharp decrease of Miss Mercury concentrations in both rice grains and also in soils. And we also see an increase of selenium concentration in the plants after we add the selenium into the soils. So that gives us a very clear evidence that selenium could inhibit the Miss Mercury accumulation in rice, and especially in rice grains. However, if we use selenium application on the leaves, which is called foliar application of selenium, we did see the increase of selenium concentration in the rice plants, but there is no change of Miss Mercury concentration in the rice grains and no change of Miss Mercury concentration in the soils. So that means the interactions between selenium and Miss Mercury within the rice plants should be less important for controlling the Miss Mercury selenium antagonism in soil rice systems. And our further evidence show that there is a very interesting relationship between the increase of soil selenium concentrations and the decrease of Miss Mercury concentrations in soils or in rice grains. And also we found a one-to-one relationship between the change of Miss Mercury concentration in soils and the change of Miss Mercury concentration in rice grains. So just think about this relationship. So this may be a little complicated to understand. So maybe you can spend 1 minute to look at this relationship and think about what these figures told us. And also think about the difference, what we showed before, between the foliar application of selenium and the soil application of selenium. So maybe you can think about that. This relationship will tell us that the decrease of Miss Mercury concentration in the rice grain under selenium amendment is mainly due to the decrease of Miss Mercury concentration in the soils. So that means the antagonism or the interactions between mercury and selenium in the soils is mainly responsible for the Miss Mercury selenium antagonism in soil rice systems. And what's the reason for these interactions between mercury and selenium in the soils? And we used some instrumental analysis, including TEM-EDX and EXINS analysis, which shows the speciation of mercury in the soils. And the results of this analysis show that after we add the selenium into the soils, there is some very evident change of mercury speciation in the soils. In the presence of selenium in the soil, the mercury speciation in the soils changed from mercury sulfide to mercury selenium, which is highly refractory and is not available to the microorganisms which are responsible for the methylation process. So that means the changes in mercury speciation under selenium amendment in soils just make the mercury less bioavailable to the bacteria. And in fact, we also detected the mercury selenium nanoparticles in the soil after we added selenium into the soils. And this could be the main reason of the decreased Miss Mercury concentration in soils under selenium amendment. So now we can have a very short summary of the story about using selenium to reduce mercury availability in soils. So first, let's look at the left panel of this figure. So after selenium enter into the soils, there will be a very quick transformation of selenium speciation, and there will be a formation of mercury selenium complexes. Actually, it's the mercury selenium nanoparticles, which has very low availability to bacteria, and this will decrease or reduce the mercury methylation process and decrease the soil Miss Mercury concentrations under selenium amendment. And this process we called Miss Mercury selenium antagonism in soil. And consequently, this will decrease the Miss Mercury uptake by the rice plant, and finally, the Miss Mercury concentrations in the rice grains. And this could be the main reason for the reduced green Miss Mercury concentrations under selenium amendment. However, if the selenium is applied on the leaves of the plant, the selenium can also enter into the plants, but there could be no relationship, no interactions between the selenium and Miss Mercury within the rice plants. And so under that way, there is no change of green Miss Mercury concentrations under selenium amendment, although the selenium concentrations in the rice tissues also increase on that. So now we can see that the underlying mechanism of the Miss Mercury selenium antagonism in soil rice systems is mainly because of the interactions of Miss Mercury and selenium in the soils and the reduced mercury availability to bacteria under selenium amendment. So maybe we can spend 1 minute to look at this figure to digest this information. So if you have any questions, you can just send me an email, or you can just text me, or you can just ask me questions after the lecture. So I'm very happy to answer your question. So this is the first example about how we can just reduce the mercury bioavailability to bacteria and then reduce the Miss Mercury bioaccumulation in crops or rice plants. So this could be a very environmental friendly method to remediate the Miss Mercury or the mercury in soils, because you know that in many countries in the world, selenium is used as a fertilizer because of the deficiency of selenium in soils and also in the food. So that means people need more selenium in their food. So that is why the selenium fertilizer are highly encouraged to be applied into the farming soils in many parts of the world, including in China. It has been reported that about, I can't remember exactly, maybe 30% or 40% of the food in China has some kind of deficiency in selenium. So if you can just use the selenium fertilizer and just apply it into the soils, this will at the same time increase the selenium concentration in the food. And of course, the farmers are happy because they can sell it at a higher price compared to normal rice grains. And the second, it can reduce the mixed mercury accumulation in the rice plants in some places, especially in those mercury contaminated areas. So maybe you may have a question. If these farming soils are contaminated, for example, by mercury or for example, by other metals, why we just abandon these farming soils? We do not use it, then there will be no problem for food safety. However, you should understand that in some Asian countries, the farming soils is not enough to support the food of the country. So that means in some countries, even the contaminated farming soils has to be used for agriculture. So that means they cannot just move outside of these contaminated areas. They may still need to use it for agriculture. So in that way, we have to remediate these metal contaminated areas. So this is the first example about how we can control the mercury bioavailability and remediate the mercury contaminated soils. And our second example is to reduce the availability of mixed mercury to the plants. So that will subsequently reduce the bioaccumulation of mixed mercury. So in that case, there's one special thing we can use, which is called biochar. Actually, there are increasing interest about using biochar to immobilize metals in the soils or in the sediments. Unfortunately, very few studies have ever considered biochar to remediate the mercury-contaminated soils or sediments. I cannot tell you the reasons for that, but it's true. So if you search Web of Science for the keywords including biochar and mercury, actually there are only about like 20 or 30 papers you can find. And many of them just use biochar to absorb the gases mercury in the air instead of mercury in the soils or sediments. So definitely the studies about the interactions between biochar and mercury in the terrestrial system and especially in the farming soils are lacking. So some pioneering studies showed that biochar could reduce the accumulation of both inorganic mercury and mixed mercury in the crops. However, the underlying mechanism of this is not clear at all. So for example, some studies in 2016 show that after we add about like 1% of biochar, I mean the soil mass, 1% of the soil mass of biochar into the soils, there will be a very sharp decrease of mixed mercury concentrations in the rice grains, as you can see from here. So only 1 to 4% of biochar applied into the soils will result in more than 90% of decrease of mixed mercury concentrations in the rice grains. And also it will result in 98% of bioavailable, or you can say phytoavailable mixed mercury in the soils. So that means biochar is very efficient to reduce the bioavailability of mixed mercury in the soils and also the accumulation of mixed mercury in the rice plants. So this is good because the price for biochar is decreasing all the time with the improvement of techniques. And it tells us we can use biochar as a candidate to remediate the mercury contaminated soils or sediments. But how about the mechanisms? If we cannot understand the underlying mechanisms of how biochar could interact with mercury in the soil rice systems, we cannot use it because it's not dependable. So normally, what people think about the interactions between biochar and mercury in soils and sediment, as we can see from these several publications, is that they believe that biochar have some very strong binding sites for mercury. For example, it has some silyl groups which bind strongly with mercury in the environment, for example, in the soils or sediments. So this will immobilize mercury in the soils or sediments and will reduce the bioavailability of mercury in the soils or sediments and will also reduce the bioaccumulation. However, some recent studies show that this may not be right for mercury. Some studies show that instead of the direct binding between biochar and mercury in the soils or sediments, there are some kind of indirect effects of biochar on mixed mercury bioavailability. Those pioneer studies show that biochar could increase the mixed mercury binding size in soils instead of itself. So that means the biochar could indirectly increase the mixed mercury binding size in the soils, and that result the strong binding of mixed mercury in the soils instead of in the biochar itself. And this indirect effect will result in the evident decrease of mixed mercury bioavailability in soils under biochar amendment. And some pioneer studies show that the contribution of this indirect effect is more than 70%. And the direct effect of biochar binding with mixed mercury is less than 30% of the contribution of the reduced mixed mercury bioavailability. So this will give us some new idea about the interactions between biochar and metals in the soil systems. So biochar could have so many kinds of indirect effects on metals or the dynamics of metals in the soils. Maybe not only for mercury and also for some other metals. And besides, some studies also showing that biochar could have very evident effects on the microbial communities in the soils, and which will subsequently change the metal speciation in the soils and also the bioavailability of metals in the soils. So maybe we can think about what we can do in the future in these aspects. So this is the second choice about how we can reduce the mixed mercury bioavailability in soils by using the biochar amendment, which will subsequently reduce mixed mercury bioaccumulation in the rice plants. So now we have a very, very definite answer for the choice 2. The answer is yes, we can control the mercury bioavailability, and in this way we can remediate the mercury contaminated soils. So the advantage of this method compared to the heating method, as I introduced before, to heat the soils to about 800 degrees or 700 degrees and eliminate the mercury in the soil, is first, the cost is much lower. So we have calculated the cost of heating and the cost of immobilizing or reducing the mercury bioavailability in soils. So as you can see from this figure, the cost of heating is much, much higher compared to the second choice. So of course, the second method is more economic and will be more easily accepted by the farmers or by the government. As a second, think about environment friendly. So this is the place where you can just get the soil out of the paddy soil and heat it using the machine I showed before. So definitely it's not environment friendly at all, because it will just destroy the farming system in this area by heating the soils to 800 degrees. And this is the place where we use the second choice to remediate the mercury contaminated farming soils. We just add for example, the selenium or the biochar into the soils and it will just immobilize the mercury in the soils, reduce its bioavailability, and also reduce its bioaccumulation in the systems. So of course, this second choice is more environment friendly. And of course, it will be more easily accepted by the farmers. Nobody wants to destroy their farming soils and cannot use it for 10 years. So the third thing is about efficiency. As we showed before, that if we use biochar, it could decrease the mixed-mercury concentrations in the grass greens and also evidently decrease the phytoavailability of mixed-mercury in the soils with very low cost. So the efficiency of the second choice could be much higher, especially we mainly concerned about mixed-mercury instead of total mercury in the environment. So that's all for the introduction of this lecture, and these are some literatures that you may read after this lecture. Thank you. And this is my email, so you can contact me if you have any questions. And then I want to let the rest of the time for the students to ask questions, and we can just communicate to each other. Thank you.
Thank you very much. That's a really clear presentation, and very interesting, the different approaches that you can take to remediate mercury, and quite a contrast in comparison with the traditional methods with the soil heating compared with the new methods that you're able to tell us about today. And I just wanted to start by asking a question about the selenium. And you mentioned that there is an issue with selenium deficiency, but the levels that you have to add, is there any risk of excess selenium for human health? And secondly, is this a one-off treatment, or is it something that has to be repeated at various intervals of time?
Thank you, Margaret, for the question. Actually, this is a very good question, because we all know that there is a very narrow window for the selenium deficiency and selenium toxicity. That means a little higher concentration. Oh, sorry, I have some screen protector that changed the color of my screen. So now it's okay. Sorry. So what we should be very careful about selenium application into soils is to control the selenium concentrations in the soils. So definitely in all the countries, there is some limit for the selenium application into soils. For example, normally what we add into the soil is about like less than 3 milligram per kilogram of selenium. I mean the total selenium concentration in the soils. So that will guarantee that there's no toxicity of selenium to the plants or to the human beings or to other organisms. So this is one thing we should be very careful about. And second, your second question is about, so that means the duration that the selenium can control the mercury bioavailability, is that right?
Mm-hmm. Just, do you have to apply it more than one time? Is it something that you have to do every year or something like that?
This is a very interesting question. Actually, we did that for about 3 to 4 years. Mm-hmm. And we found that after one time of application of selenium, we can still find the evident decrease of mercury bioavailability after 3 to 4 years. Mm-hmm. So we haven't done a much longer time, but 3 to 4 years is okay. That means one single application, and it can save you. You can protect from mercury problem for like 3 to 4 years. So that also means the cost of this method is pretty low compared to other methods. Mm-hmm. And it's also same for the biochar application. So we have found that for 3 years, we found exactly the same decrease in mixed mercury concentrations in rice grains following biochar amendment. That means just one time of biochar application, we found a decrease in mixed mercury bioavailability and bioaccumulation. Mm-hmm. So that's good news.
Yes, yes. Are there other questions?
Well, and just like there are different types of different forms of mercury, there are different forms of selenium as well. Does it matter what the selenium form is that you add?
Yes, this is a very interesting question because actually we try different species of selenium. And this is also true for the selenium fertilizer farmers use. They also use different species of selenium. But the good thing about selenium is that there is very quick transformation of selenium species in the soils. As shown by our experiment, the transformation of selenium species is within days. So that means no matter what species of selenium, for example, selenite or selenate, you add it into the soils, the effects are exactly the same to reduce the mixed mercury bioavailability and bioaccumulation. So that's also a good news about selenium application. So is there any other questions from students?
I'm not seeing any. You mentioned that you know, these are important farming. Oh, there's women. Yeah, I can see a question here. Can you see the question from Michael?
Let me see. So the question is the selenium mercury compound stable under different environment conditions? For example, if the paddy field will dry out. So this is the question.
Mm-hmm.
So the question is that, is the mercury selenium complexes stable in different environments, especially on the oxide condition?
Mm-hmm.
So actually we tried it. And generally speaking, this complex, I mean the mercury selenium complexes, is very, very stable, and is more stable compared to the mercury sulfide complexes.
Mm-hmm.
And this is why we found that there is a speciation change from mercury sulfide to mercury selenium complexes. So the answer is yes, it's very stable. But if you consider the potential change in the mercury concentration under anoxide and oxide condition, for example, in a rice wheat rotation system, which is very common in Asian countries. We haven't checked the speciation change in such kind of system, and we cannot tell exactly if there's how many percentage of change about mercury concentration. So you know that mercury selenium or mercury sulfide is not the only species in the soil. So the answer is we don't know. We don't know what's the speciation change in under anoxide and oxide condition, but definitely there will be some kind of change in mercury speciation. But the good thing is that the mercury selenium complex is very, very stable. So I think that's the answer to the question. I don't know whether it's clear enough.
Yeah, and I was going to ask you, you mentioned about the farming soils, you know, and the importance of these, that you can't just go somewhere else. Do you have a feel for the proportion of the farming soils that are sufficiently contaminated that they need to be treated?
Sorry, can you just repeat your question again? I cannot hear very clearly.
Yeah. Do you have an idea of what proportion of farming soils need to be remediated? Is it 10%, 50% of the farming soils?
You mean in China or
in China? In China, yeah. Mm-hmm.
So my direct answer to this question is that it's not my responsibility to answer this question. It's not my ability to answer this question because we need a national-wide survey about the contamination of farming soil in China. But according to the official data in China, about 1.6% of the soils in China has some kind of mercury contamination, but that includes also farming soils or non-farming soils. Mm-hmm. So if your question is to ask at what concentration we should try some remediation to guarantee the food safety. So maybe we can answer that after a series of experiments with different concentrations of soils in different parts of China. And that may be a very interesting project to work with. Thank you for the question, Margaret.
Yeah. And your work, I mean, I saw the graphs that said brown rice. Have you looked at different rice types and any variation in the bioavailability of the mercury to different types of rice? Does your method work equally well for all rice types?
You mean our remediation method?
Yeah. Mm-hmm.
The answer is no, because we haven't tried to verify our method to different varieties of rice, but we have tried different type of soils.
Mm-hmm.
That's more important, because what we think is to control the availability or the mobility of Miss Mercury in the soils to all kinds of plants, including rice or wheat or some other crops. But another interesting story I would like to tell is about the difference in different variety of rice. Actually, we tried about like 20 or 30 different varieties of rice, and we found that there's a huge difference in mercury accumulation in this different variety of rice.
Mm-hmm.
The first we think that is because of the difference in availability of Miss Mercury. But what surprised us is that we found that the rice itself could also affect the Miss Mercury concentrations in soils. That will subsequently affect the Miss Mercury accumulation by rice itself. And one of my students is doing some kind of research which showed that the root exudate, that means the organic matter extracted by the produced by the root, will affect the Miss Mercury production process or the microbial mesmerization process in the soils, and will have huge impacts on the soil Miss Mercury concentrations. And the next step, we want to analyze what's the components in this root exudate, and tell what's the major components in this root exudate that controls the mercury methylation process in the soils. So this is a very interesting story, because the plants itself could also affect the Miss Mercury concentration in the environment and subsequently affect itself. So I don't know whether it's because of some kind of evaluation, some kind of a change in plant physiology or something.
Mm-hmm.
So that's a new story. Maybe we can tell
Yes.
it next time.
Yes, yes. We got any more questions from others who are online? Okay, I'm not seeing any more. Can I just ask a little bit about your biochar? Because we've worked a little bit on biochar, and I just wondered if you had any views as to the materials that you make the biochar from, whether it must be virgin biomass, or whether you can make the biochar from waste materials, in terms of your ability to then add the char to soils that are used for producing food crops. Does it matter the source of the char?
So this is a very interesting question. It's just like you are just working in my lab, because that is what we are doing now.
Mm-hmm.
Actually, one of my students is working on different materials that produce the biochar. We try different crop straw. And some other colleagues in other universities also tried some kind of sewage waste and to produce the biochar, and to see what's the different effects of this biochar is produced from different materials, and what's the effects on the metal mobility in the soils or sediments. And what's interesting about mercury is that biochar could increase the phytoavailable or the bioavailable Miss Mercury concentration in soils for some kind of biochar, but decrease it for some kind of biochar. And the reason is we cannot tell all the reasons for that, but one thing very special is that the sulfate concentration in the biochar, because the mercury methylation process is very sensitive to the sulfate. So first, the sulfate could affect the activity of sulfur-reducing bacteria, which is mainly responsible for the mercury methylation process. And the second, the reduction of sulfate to some reduced sulfur species could immobilize inorganic mercury in soils and also miss mercury in soils. That will affect the bioavailability of mercury in the soils. So in these two aspects, the sulfate concentration in the biochar could affect the mercury bioavailability. I think it could be also true for some other metals because of the controls of those reduced sulfur species on metal mobility or bioavailability. So this is one thing special about this. And some others also consider like silicon or some other components within the biochar. But I'm not so familiar with those factors. Yeah, thank you for the question.
That's all right. Okay, so if there are no further questions just now, I think we can finish the session. If anyone wants to ask more questions from Huan, there's email addresses here, and it'd be great to get in touch and find out more about what he's doing. But for now, I'll just say thank you very much for an excellent presentation. I really enjoyed listening to this about the mercury and also about the biochar as well, because we're also working a bit in biochar. But thank you for just now, and I think we should log off and let you enjoy the rest of your evening.
Thank you. Yeah, thanks for listening. Hope that if you have any question, just contact me.
Yeah. Okay. Thank you. Bye.
Bye.